All right, you guys are going to start off with today is sponge anatomy. So we're going to break this into two chunks. First, we're going to look at the sponge as a whole and the direction of water flow, and then we're going to go ahead and talk about those specialized cells inside. So really quick, what you're looking at here is a very generic picture of a sponge, and what you're seeing here is as if the sponge were cut so that you could see inside of it. So the central cavity here is also called the atrium, and that's the middle chamber of the sponge. You can see here direction of water flow. Water flows through the holes in the sides of the sponge and then out the osculum. All right, so the holes in the side are called the ostia, and the bigger hole at the top where water leaves is the osculum. All right, if you follow this box right here, this is an enlarged view. Follow the green arrow. This is an enlarged view of that box. So you can see the actual cells that are involved here that make up the wall of the sponge. So this is, these are the collar cells. We have the amoebocytes. You'll notice the amoebocyte and the collar cell are right next to each other. We're going to talk about why that is in just a minute. And then we have the pore here, which again is also called the ostia. Follow the green arrow down again. We're going to take a bigger view of the collar cell. You have the flagellum, which creates water movement, the collar itself, which traps the food, and then this is the body of the cell that is going to move food to the amoebocyte. Oops, sorry. So take a look here. Actually, go back. We're going to go back to this one real quick. Before we leave this permanently, go ahead and pause the video right now and copy this basic diagram. You don't need all of the immense detail, but copy this diagram to show water flow and label the ostia and the osculum here. All right, once you're done with that diagram, we're going to talk, this is a more detailed view of what you just saw. So again, the blue arrows are representing the direction of water flow. But what I want you guys to look at here is the connection between the collar cell and the amoebocyte. So again, food particles are collected here at the collar and moved inside the cell through a process called phagocytosis. This is literally just the cell itself is absorbing the food in these little tiny pockets, and then those little tiny pockets end up getting transferred to the amoebocyte. The amoebocyte is a specialized cell that takes nutrients all around the rest of the body of the sponge. So take a look here. We have all the collar cells. Notice where they are. They're lining the insides of the sponge, creating that water movement, right? The pores are also all along the sides of the sponge, allowing food particles in, and then the amoebocytes are connected to them. To the collar cells, and the amoebocytes are going to make sure that all of the other pieces of the sponge, the skin cells, the epithelium, have appropriate nutrients, and that everybody has what they need in order to survive. Very key there. If you would like, go ahead and draw a detailed view of the collar cell with these labels. And then when you're ready, go ahead and pause if you need an extra minute, and then when you're ready, go ahead and start again. All right, so specialized cells. There are two major types of specialized cells in the sponge. The first is the collar cell, which we've talked about, and the second is the amoebocyte, which we have also talked about. The collar cell, again, it has a couple major jobs. It's to create water flow in the sponge by beating the flagellum. And then it is also meant to catch food particles. So again, here is your choanocyte, your collar cell. And notice again, they're lining the internal cavity of the sponge. The amoebocyte, again, is to pick up nutrients and distribute that to the rest of the cell. Here is a quick animation. If you look at the right, this is an up-close view of how the collar cell works. You can see the food particles making their way in and making their way inside here. And then again, the amoebocyte would be on the edge of the collar cell collecting the food. What you see on the left here, this is how sponges get their oxygen and their food. You can see the flagella are beating in sequence, to all coordinated. That's very important to create the water flow in the correct direction. We want water to go from the outside of the sponge into the atrium or the inside of the sponge. Sponges also have some other important structures. We've already talked about the pore cell, but the pore cell is a cell that creates the ostia, right? So it is a cylindrical cell. It has a hole in the middle, and that's what creates the holes in all the sides of the sponge. We also have the osculum, which is the larger pore that lets water out. And then we have these, we have the spicules, which are glass-like structures. You can see them right here. The glass-like structures, they are in a couple different shapes, and what they do is they interlock and create a support or a skeleton for the sponge. There's one other important structure. It's called spongin, and spongin is essentially what makes a sponge spongy. It's the squishy stuff in a sponge. The spongin and the spicules together typically make up the skeleton of the sponge. All right, so we're going to go ahead and stop at this point today. I'm going to have you guys move on to your first activity, which is an anatomy practice activity. You're going to practice just labeling the important parts of the sponge, and then there will be a quick quiz on teams that you should access. So this lecture has 2 assignments associated with it. One is on paper that you should get from the substitute, and the other is on Teams, and it is a multiple choice quiz. If you have any questions, you can send me a message on Teams, and I'll respond to you sometime later today.